Production of vinegar. Vinegar is a preparation containing acetic acid. So we can call this as acetic acid fermentation also. But there is a difference between acetic acid, pure, and vinegar, because there is a definition of vinegar. Vinegar is a mixture, and acetic acid, when we use the term acetic acid, we use it to describe pure acetic acid, and vinegar is the solution. So the definition of vinegar is the preparation that contains acetic acid with minimum 4 gram percent. This preparation is called as vinegar, and this vinegar uh, has a use as a flavoring agent in food. In French words, vinegar means sour wine, so it indicates that the raw material for the preparation of vinegar is wine. So wine, when it is sour, it is called as vinegar. That means when microbes present in wine converts that ethanol to acetic acid, it becomes sour wine. So it involves acetic acid fermentation. Now vinegar is a preparation that has this following composition. It means that it does not have only acetic acid, it has several other components. So it contains acetic acid, small quantities of glycerol, which is responsible for giving sweetness, organic phosphates, organic acids like pyruvic acid, formic acid, propionic acid, and butyric acid, and several other products of microbial respiration. It includes diacetyls, acetone, isobutyraldehyde, isovaleraldehyde, a methyl valeraldehyde, methyl isobutyl ketone, and esters of methyl, ethyl, isobutyl, and isoamyl alcohols. Free propyl, secondary butyl, isobutyl, and amyl alcohols have been also reported in vinegar. So in the high concentration there is presence of acetic acid, and along with that the other components are also present. So the distinct flavor of vinegar, it is because of the particular combination of these substances and not from the only acetic acid. There are different categories of vinegars which are distinguished on the basis of the type of raw material which is used for the making of alcoholic broth. So we know that for production of vinegar, the raw material or the medium required is alcoholic broth, that means which contains ethanol. So depending on the source from where this alcoholic broth is obtained, there are different categories of vinegar. Now this you need to understand very carefully, which is important in relation to MCQs. Apple vinegar, this is the product of alcoholic fermentation and subsequent acetous fermentation of juice of apples. Then wine vinegar, this is a product of alcoholic fermentation and subsequent acidus fermentation of juice of grapes. Malt vinegar, this is a product of alcoholic fermentation and subsequent acidus fermentation of barley malt or cereals. Sugar vinegar, this is the product of alcoholic fermentation and subsequent acidus fermentation of sugar syrup or molasses. Glucose vinegar, it is a product of alcoholic fermentation and subsequent acetous fermentation of solution of glucose. And the last one, spirit vinegar. This is the product of acetous fermentation of dilute distilled alcohol. Now the method of production of vinegar. It involves the souring of alcoholic broth under controlled conditions. Production of vinegar, it is a two-stage reaction or two-stage fermentation process. In the first stage, the alcoholic fermentation happens, and in the second stage, acetic acid fermentation happens. So in alcoholic fermentation, during this stage, the sugar containing raw material, that is fruit juices, honey, molasses, hydrolyzed starchy materials, are fermented to ethanol fermentation. That means this is ethanol fermentation, which we have already studied in earlier chapter, by a selected strain of yeast. So this part we have already studied. This is ethanol fermentation, and after the ethanol fermentation is over, we have alcoholic broth, and that alcoholic broth is ready for acetic acid fermentation. So this alcoholic fermentation, it involves the similar events which we have already studied. The production medium should contain a sugar concentration approximately from 10 to 20 depending on the strain of Saccharomyces used, plus other ingredients. The fermentation is carried out at desirable temperature and pH for a specified incubation period. Alcoholic broth is then withdrawn and it is separated from the yeast cells, and then this alcoholic broth is used as a raw material for acetic acid fermentation. Now what happens in second half of fermentation, that is acetous fermentation or acetic acid fermentation? The conversion of ethanol to acetic acid, it is brought about by aerobic acetic acid bacteria. This is very important, which are Acetobacter acetogenum, Acetobacter pascuranium, Acetobacter xylenum, etc. So this is an aerobic fermentation, and this process requires large quantity of oxygen because this is oxidation process. Ethanol is oxidized to acetic acid. Say for example, the conversion of 1 liter of ethanol, that is alcohol, to acetic acid, it requires 552 grams of oxygen. So this oxidation process, it releases considerable heat. That is a feature of this fermentation. Therefore, there is a requirement of well equipped fermenter which has a facility of aeration and a temperature control for the effective conversion. So there are several methods of microbial conversion of dilute ethanolic solution to acetic acid. The first method of vinegar production, which is the one of the old methods, is Orleans process. This is the old process. Nowadays it may not be used. This is the old French process in which acetic acid bacteria grow slowly as a film over the surface of still liquid, means a static liquid, and this process is slow. This process involves the use of barrels which are made up with oak. So oak barrels are useful for the storage of wine. So these barrels of approximately 200 liters capacity were filled one third full with vinegar and the starter culture bacteria. So this is the mixing solution. It contains vinegar itself plus starter culture bacteria and 10 to 15 liters of wine. That is, this is alcoholic broth, this is vinegar, This is starter culture. So at weekly intervals, the same amount of wine is added to the barrels. And after 5 weeks, 10 to 15 liters of vinegar is withdrawn. So there is addition of wine, and there is withdrawal of vinegar at regular intervals, and this process, it is repeated several times. So the bacterial slime, it grows on the wooden gratings or the crossbars, so that facilitates the immobilization of acetobacter cells. This is oak barrel, and these are the wooden gratings or the wooden support, and this is for the passage of air. And this is a mixture which contains vinegar for acidity, then wine, and then there is uh, a wooden grating, and then there is addition of culture. So what happens after addition of wine? The bacterial slime will develop, that means the acetobacter cells will grow on these wooden gratings. So these cells will be immobilized on these wooden gratings, and they will come in contact with alcoholic broth, and it will be oxidized in presence of air to acetic acid. This is called as Orleans process.